I'm Trudi Schupbach. I'm a Howard Hughes investigator and the professor of molecular biology at Princeton University. In my previous segments of my talk, I have introduced you to the question of pattern and polarity during Drosophila oogenesis. I've also told you that there is a key signaling process that takes place during oogenesis, whereby a signaling molecule encoded by the gene Gurken signals to the Drosophila EGF receptor, which is expressed in the follicle cells, and induces dorso-ventral pattern formation in the follicle cells. The localization of the Gurken protein depends, as I've told you in the second part of my talk, on the localization of the Gurken RNA. Once the RNA is localized and the protein is made, it is then released from the oocyte and diffuses through the space between the oocyte and the overlying follicle cells, binds to the EGF receptor, and starts this pattern formation process. In the third part of my talk, I would actually like to start telling you about some of the experiments we are doing to try to understand what the response process is that goes on in the follicle cells, and also how the Gurken gradient actually shapes this response in the follicle cells. Just to recap what I've already told you, so the Gurken RNA is transcribed during oogenesis, and then there is a set of proteins that are involved in the localization of the RNA to the dorsal anterior corner. There is also translational regulation of the Gurken RNA, and once it's localized, the protein is made and then secreted by the oocyte, binds to the EGF receptor in the follicle cells, and this binding activates the receptor and activates the downstream signaling process that then results in the establishment of dorsal follicle cell fate. One of the outcomes of this activity of the EGF receptor in the dorsal follicle cells is to repress a new signal that is created by the follicle cells and that is transmitted back to the egg and then serves to establish the dorso-ventral pattern of the embryo. So ultimately, what starts in oogenesis with the movement of the oocyte nucleus ends during embryogenesis with the formation of a gradient of a protein in the embryo, a protein that is called dorsal. The dorsal protein accumulates to very high levels in the nuclei on the ventral side of the embryo. And it is mostly cytoplasmic and not in the nucleus, on the dorsal side of the embryo. Dorsal encodes a transcriptional regulator, and once it's in the nuclei of the ventral embryonic cells, it regulates transcription in these cells and thus establishes ventral cell fates. Now, during my talk before, I've told you that during oogenesis, we have a signaling process, Gurken EGF receptor, going on the dorsal side of the egg chamber. So how does this all hang together? So during oogenesis, we have Gurken localized to the dorsal side. It activates the EGF receptor on the dorsal side, and that then restricts a new signaling process to the ventral side of the follicle cell epithelium. This new signaling process requires the activity of a gene called PIPE. And the PIPE gene starts this new ventral signal by activating a protein called Spatial, which can bind to a receptor that's present on the egg plasma membrane. And once this receptor is now activated on the ventral side, it leads to the import of this dorsal transcription factor into the nuclei on the ventral side and the formation of this nuclear gradient of the dorsal protein. So the effect of Gurken EGF receptor signaling on PIPE expression is shown here in this slide. taken by the laboratory of David Stein, where he showed here in the wild type that pipe expression is seen on the ventral side of the egg chamber, whereas in a Gurken mutant, so when Gurken doesn't signal, the EGF receptor is not activated, and now pipe is expressed all throughout the egg chamber. So pipe is then a target gene of the EGF receptor and is repressed by EGF receptor activity. So what we are then interested in understanding is how does the EGF receptor affect the expression of pipe? Is this a direct repression in response to EGF receptor activity, or are there many steps in between? Are there other signaling processes that intervene and that eventually regulate the expression of pipe and restrict it to the ventral side of the egg chamber? In order to get at these questions, we have again undertaken some mutagenesis experiments to find genes that would alter EGF receptor signaling or its outcome in the embryo, to try to understand how this process works at the molecular level. In these mutagenesis screens, we used a technique that allows us to make mosaic egg chambers. We suspect that many of the genes that might intervene here downstream of EGF receptor signaling might be lethal for the homozygous carrier, and for that reason we can't just simply do mutagenesis and look at homozygous mutant flies, but we have to work with heterozygous flies that can survive and then induce homozygous mutant follicle cells inside these heterozygous flies. The way we can do this in flies is by using a system that allows us to generate mitotic recombination. So normally during mitosis, of course, there's no recombination, but by introducing homologous recombination enzymes and target sites from yeast, fly biologists have engineered chromosomes that can now be used to make such homozygous mutant cells within a heterozygous carrier fly. Initially, this happens at mitosis. A single cell will be homozygous mutant, but then as the cell divides, it will form a patch or a clone of cells that are all homozygous mutant. What this looks like is illustrated here in this slide, where you can see we made a patch of mutant follicle cells. These follicle cells were mutant for a mutation that makes a very thin eggshell. So you can see here, when we use the technique, we create a mosaic eggshell where we just have a large patch of mutant cells surrounded and embedded in normal wild-type follicle cells. A postdoc in the lab, Li Mei Pai, performed such a screen, and she found a number of mutations that showed interesting phenotypes. These phenotypes here can be classified as dorsalized eggshells, and the embryos that developed inside these eggs were also partially dorsalized. Li Mei then analyzed these mutations at the molecular level, and she found that, for instance, the one here shown on the top is a mutation in a Drosophila gene called Sybil, which is a conserved gene that's also found in vertebrates. It has a C. elegans homolog, and then two different protein isoforms are made from this gene in flies by differential splicing. What does the Sybil gene do? Well, about 10 years ago, there were a series of biologists that investigated this Sybil gene because it is actually an important oncogene that can be implicated in cancer formation and mutant in humans. And they found that the Sybil genes can act to downregulate the activity of major receptor tyrosine kinases, such as the EGF receptor. So normally, when these receptor tyrosine kinases are activated, because they bind their ligands, they cross-phosphorylate, and once they are phosphorylated, they are active and now can also phosphorylate the target proteins inside the cell. What Spatial does is it has a protein phosphotyrosine binding domain, and it binds to these activated tyrosines, phosphorylated tyrosines in the receptor, and then acts as a ubiquitin ligase to get the receptor marked with ubiquitin. This serves both as a targeting signal to downregulate the receptor and also as a degradation signal to get the receptor degraded. So the Spatial proteins have therefore an important function in limiting the activity of the receptor. So that makes now sense with respect to the phenotypes that Limay saw in these mutant egg chambers. In fact, we now can understand that normally the EGF receptor has to be active to make us a dorsal side, but then if it's overly active or stays active too long, this will result in dorsalized eggs and embryos. Now we wanted to really understand better, however, whether Spatial is really only required on the dorsal side to downregulate the receptor, or whether it has any effects on the ventral side, and how, in general, it shapes the activity of the receptor. For this, we used some target genes of the EGF receptor to see how they would be misregulated by the loss of Spatial function. CHECON is one of such EGF receptor response genes. It usually is activated by receptor activity, and therefore at this stage of oogenesis you can see that there is a high level of CHECON expression on the dorsal side of the egg chamber in response to EGF receptor activity. Now, when Limay made clones, mutant clones that had lost Spatial activity on the ventral side of the egg chamber, and they're shown here in black. So the wild-type cells in this egg chambers are labeled in red, but as they became mutants, they lost the red marker, and that tells us that they are homozygous mutant for Spatial. But interestingly, in these ventral cells that are mutant for Spatial, you can now see high CHECON expression on the ventral side, where it should normally not be expressed. That tells us that Spatial must be acting in general to keep the EGF receptor activity low on the ventral side, and that if Spatial doesn't act, EGF receptor activity gets high on the ventral side and activates a dorsal response gene. Interestingly, also, if you look very carefully, you can see that the expression of CHECON, the misexpression of CHECON, is very cell autonomous. It just happens in the mutant cells, not in the neighboring cells. That tells us then that the activation of CHECON by EGF receptor is very direct. It happens, each cell in a way looks at its own EGF receptor activity and decides whether or not to turn the response gene CHECON on or not. It doesn't look at its neighboring cells. It just measures somehow the level of EGF receptor activity inside its own cytoplasm and then turns on the response gene cell autonomously. We can look at another response gene, PIPE, and we find the same situation there as well. Now, PIPE is repressed by EGF receptor activity, and you can see in wild type, and this egg is a little bit tilted, so we're looking mostly at the ventral side, but you can see it has a sharp on-off expression pattern. It's off on the dorsal side, it's on the ventral side. And if we make a clone of mutant cells for Spatial here on the ventral side, PIPE gets repressed. Again, telling us that Sybil normally has to keep EGF receptor activity under control on the ventral side in order to allow pipe to be expressed. Not shown in this slide, but also done in experiments by Li-Mei, is that she was able to show that all of this Sybil activity depends entirely on the presence of Gurken. So in a Gurken mutant, pipe is just simply expressed everywhere, whether or not Sybil is there or not, telling us therefore that what we're looking at here really is the result of Gurken EGF receptor signaling and not some other signaling pathway that's affected by the Sybil mutant. These results, though, then led us to revise our view of the Gurken gradient to some extent. The fact that in the absence of Sybil, Gurken can activate the EGF receptor even in ventral follicle cells tells us that some Gurken protein must actually reach the ventral side. So shown here in this cartoon form, Gurken RNA is localized to the dorsal side of the egg chamber in a very sharp domain. But then the protein that's released from the egg must be able to diffuse to a certain extent to the ventral side. Under normal circumstances, Sybil is there to help keep EGF receptor activity low on the ventral side, so that these occasional Gurken molecules that do make it to the ventral side don't have the effect of repressing pipe or activating Kekon under normal circumstances. So in the wild type, we see Kekon only expressed on the dorsal side, where we have high EGF receptor activity, and pipe expressed on the ventral side, where there is low EGF receptor activity. But then in the Sybil mutant, where EGF receptor activity overall gets increased, now Kekon can be expressed from dorsal to ventral, and pipe gets repressed even on the ventral side. We concluded that Gurken must reach the ventral side by doing these mosaic experiments and looking at the expression of the response genes. Meanwhile, Li-Mei Pai in her own laboratory has gone on and she has used a system where she can visualize Gurken molecules during oogenesis much better than we could with the regular antibody, and she has in fact found that indeed she can see some Gurken molecules in the follicle cells on the ventral side of the egg chamber. However, we now have a new question. So the Gurken gradient is established and it reaches from the dorsal side all the way to the ventral side. So what are the biophysical parameters of this gradient? How can it activate response genes? What are thresholds for these response genes? How can we think about pattern formation from the dorsal all the way to the ventral side? We can use antibodies to look at the Gurken protein. Li-Mei has been able to visualize the Gurken molecule here with a fusion protein. But the problem that is vexing the field is that while we can see the Gurken protein in various stages, it's hard to know which of these molecules that we are visualizing here are actually the active protein. The protein is made as a precursor, it's cleaved and then can diffuse, and then it's taken up by the follicle cells and eventually degraded inside the follicle cells. So where are the molecules that are really actively forming the gradient? How can we think about those, or how can we get some impression of what the shape of the Gurken gradient is? In order to address this question, I have been for a while collaborating with a colleague of mine at Princeton, Stas Shvartsman. And he and his students have been modeling dorso-ventral pattern formation in oogenesis. And together we had this project where we were combining some experimental approaches with the modeling approaches to gain some description of the shape of the Gurken gradient. So how was that done? Well, Lea Guantoro, a graduate student of Stas Shvartsman's, employed both experimental manipulation of the Gurken gradient, and she also did modeling of the Gurken gradient, and then she did parameter fitting and estimations of parameters that allowed us to get some sense of the Gurken gradient. So initially, Lea had to write all the equations. She had to model the Gurken gradient. This would involve the production of the molecule, its diffusion, its binding to the receptor. The receptor, of course, has also to be made in the follicle cells and deployed on the plasma membrane. And all of this can then be summarized in a relatively complicated equation that I won't go into here. But the important thing to note here is that really none of the parameters have been determined in absolute terms, which means that this equation cannot just simply be solved by putting actual numbers into these equations. They all can only be described in a series of differential equations. So then how can we still think about this relatively complicated gradient formation? Well, there are some things that are intuitively understandable. We can think of two different extremes. We can have something that's a long-range gradient, or we could have, conversely, something that's a short-range gradient. A long-range gradient could be formed, for instance, if the molecule we're looking at has a very high diffusion rate, if it has a low binding constant to the receptor, so it diffuses long before it binds the receptor, or if it has a very long half-life relative to the space it has to travel through. And conversely, of course, a short-range gradient would be one that could be formed by molecules that diffuse very slowly, that bind with high affinity to the receptor so that they immediately bind as soon as they're released from the oocyte, or if they have a very short half-life. We can now test whether Gurken is a long or a short-range gradient by manipulating some of the parameters. And we decided to do this by changing the level of the EGF receptor in oogenesis. In flies, we have convenient systems that allow us to express target genes at different levels. It's called a Gal4 UAS system that's been imported from yeast. And by having different Gal4 transcriptional factors that drive expression of the UAS target gene at different levels in oogenesis, we could vary the amount of receptor that was expressed in the follicle cells. And then Lea used the pipe gene as a target readout for the Gurken gradient. The pipe gene was very convenient because it has a very sharp on-off response, and so it was easy to quantify this response. And you can see that with increasing receptor level, the pipe boundary was shifted. When Lea quantified everything, we saw that in fact the response to these manipulations of the system was quite noticeable. So the gradient therefore seems to be easily manipulated, which already implies that it can't be just that the Gurken molecule immediately binds to the receptor. It seems that by just changing receptor concentrations, even at small levels, we can make quite a noticeable effect in the pipe response. putting all these results together and then fitting the results to the model, Leah was able to conclude that Gurken indeed is a long-range signal. And in particular, she was able to calculate that approximately the difference between the concentration of Gurken on the dorsal side versus the ventral side is only 10-fold. She also could give approximate threshold levels to the Gurken gradient for the pipe and the Kekan response. We think this is an interesting approach that could potentially also be used to estimate other gradients in development, where it might also be hard to really visualize the active molecules, but by performing a simple manipulation such as giving extra receptor copies to the tissue and seeing whether the tissue responds and by how much, it should be possible to get similar estimates for other gradients as well. At the end now, I would like to thank everybody who's made it so far and listened to my talk on dorso-ventral pattern formation in Drosophila oogenesis. And last but not least, I need, of course, to thank all the many people in my laboratory who have contributed to this work over the years. And I also would like to thank my collaborators at Princeton, Stas Shvartsman and Liz Gavis, who have collaborated with us in very exciting new projects. And I hope we'll do more of those in the future. Thank you.